So this is part 3 of chapter 48, and we left off looking at this diagram, which has a lot of information, really good information. And we were talking about this sodium-potassium pump here and how it's responsible for maintaining that resting potential state where we have high potassium on the inside of the cell and high sodium on the outside. And what's really neat about this pump is that for every 3 sodiums that are pumped out, there are 2 potassiums pumped in at the same time. So this 2 to 3 ratio actually helps to maintain this higher positive charge on the outside of the cell as opposed to the inside. And again, remember we're talking about a slight difference in charge between the two, and you can see here the negative versus the positive. So at resting state, we have negative intracellular and positive extracellular. So these opposite charges on the opposite sides of the cell membrane mean that our cell membrane is polarized. This charge gradient is, in essence, potential energy, because we have like a battery, we have stored energy set up with this difference in charge. Now we're going to look at a quick animation from your textbook company and looking at this resting potential.
A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. The cytoplasm just inside the membrane is negative in charge, and the fluid just outside the membrane is positive. The membrane is said to be polarized. Like a flashlight battery, the difference in electrical charge carries potential energy, which can be measured as a voltage. The oscilloscope shows this charge difference, called the resting potential, in millivolts. A neuron has a resting potential of about minus 70 millivolts. What causes the resting potential?
The plasma membrane of the neuron keeps large negatively charged molecules such as proteins inside the cell. Pumps and channels in the membrane regulate the flow of positively charged sodium and potassium ions. The neuron maintains a higher concentration of sodium ions outside the cell than inside, and a higher concentration of potassium ions inside the cell than outside. In a resting neuron, most sodium channels are closed, but many potassium channels are open. As a result, very little sodium can diffuse into the cell, but potassium ions can flow freely out of the cell, adding to the positive charge outside. The sodium-potassium pump actively transports more sodium ions out of the cell than potassium ions into the cell. This contributes to the buildup of positive charge outside the cell, while keeping the concentration of sodium low and the concentration of potassium high inside the cell. The diffusion of positive potassium ions out of the cell and the large, negatively charged molecules trapped inside the cell cause the charge difference that constitutes the resting potential.
So again, in a non-stimulated neuron, we have a resting potential of about a negative 70 millivolts. And what I like about this slide is the emphasis at the bottom here that says to remember it's the direction the ions want to move that determines the polarity and this action potential energy that we have here for an action potential. It's not the actual molarity of the ions that we're talking about. So it's again this difference between the inside and the outside that is actually a concentration gradient that is setting up our resting potential. So how does a nerve impulse travel? Well, we get that stimulus. A nerve is stimulated through the dendrite. And what happens is that the dendrite actually receives a signal, it will often be something, a neurotransmitter binding to a receptor site on the dendrite, which actually sends a signal, and remember our signal transduction pathways, it will send a signal to open the sodium gates. And so that sodium channel is going to open up, and of course we have this large concentration of sodium outside the cell. And so what happens is as those sodium channels open up, they're considered gated channels. So the gate gets opened and the sodium rushes in. So this is when we have an action potential occurring. You need to remember that it takes a number of these stimuli to occur in order for the actual opening of all of these sodium channels. So essentially you can think again as there's a threshold that has to be met, like getting over the little rise in a hill, in order to get those sodium channels to open up enough to start changing the negative and positive charges across the membrane. This threshold, this number that you have to get past, is called the threshold potential. But what is going to happen is that first domino is going to go down. And you can think of it as that first sodium channel is going to start opening up, and it's going to end up changing the polarity of our membrane here. So instead of having negative like the resting state on the inside, we're going to end up with a positive because of this rush of positive sodiums into the cell. And then that again is going to make the outside relatively negative to the inside. So we're reversing the polarity of the membrane. And this is going to curve in a wave motion. As you can see here, what this is showing is that these channels are going to slowly be opening up one after another, because as the stimulus is, and this is actually how the stimulus is going to pass down the axon of a neuron, is through this motion of opening up these different sodium channels. These sodium channels are called voltage-gated channels. And so it takes this change here in this positive, at this point, it's the change in the positive that's going to, on the inside of the cell, that's actually going to cause the next set of channels to open up. And that's why they're called voltage-gated, because it's the change in voltage that is actually causing them to continue to open up down the line here. And the action potential is defined as the movement of this impulse down the neuron there. So now we have one wave traveling down a neuron. And that wave is made up of this different sodium channels opening. But immediately following that first wave, we have a second wave begin to happen. So what happens as the sodium builds up in the cell here, what happens is that those sodium channels actually start to close. And as they do, what happens is the potassium channels will begin to open up. So as this sodium is rushing in, our potassium starts to rush out, and they will diffuse out of the cell, and again reversing back at that point to having a negative on the inside and then the positive on the outside. So we start to get rid of this positive charge by now getting rid of our potassium ions. This is a diagram that shows that wave motion. So you can see here we have the action potential at this end of the neuron. So let's say they're showing it right here at the top of the axon, near the axon hillock. And then what's going to happen next, so we have an influx of sodium coming in, and then we have that changing the polarity here, so it opens up these sodium channels. And again, this positive charge in here is going to open up the next. But what's happening following right on the heels of this, now we start to have the potassium channels opening up. And as they do, we return back to our negative charge on the inside. And you can see this wave motion that will continue all the way down the axon until we end up at the axon terminal. And you can see here, once the potassium channels have opened up, then we have the membrane and the neuron returning back to its resting state or resting potential state. So a neuron can actually produce hundreds of these action potentials in just a single second. So it seems like this would be a very slow process, but in reality it is extremely fast. And the frequency of an action potential is the reflection of how strong the stimulus is. So when we say we have a strong reaction, a strong nerve impulse, it's really not that we have more sodium rushing in or more potassium rushing out. It's that we have more impulses quickly passing by. So the frequency, the number of action potentials passing along is actually how strong the stimulus or how strong the impulse is going to be. And remember, it only takes milliseconds to get a message from your brain to your fingertips, for example, to get it off something that's hot and burning. Another factor to consider when we're looking at the structure of a neuron is that the greater the diameter of the axon, the greater the speed of the action potential. So it's going to occur much more quickly the larger our neuron's axon is. So here, this is a diagram showing a action potential graph. And this is something you need to be familiar with. So really quickly, I'll try and squeeze this in here. But essentially what is occurring, and study this in your book, but you can see there's 1, 2, 3, 4, 5, 6 parts of our action potential depicted in this graph. And so the first part is our resting potential. So we have this resting potential here at about a negative 70 millivolts. And again, you know what's happening at that point. We have this negative inside the cell, positive outside, different resting potential there. The next step, step 2, is called our depolarization stage. So that's when we start to have movement on this graph. And that depolarization, what that means is that some of the sodium channels are beginning to open. So there's something there, the dendrite of the cell has received some sort of signal that's telling the neuron to start opening up its sodium channels. And so this is called the depolarization phase. We still haven't started in on the action potential. We're just now getting over this threshold. And it's not until we get up here to this negative 50 millivolts that we actually have enough of a change, enough sodium within the cell to start the actual action potential. So our next phase is going to be that third phase, which is the rising phase of the action potential. And what's occurring during that time is a lot of sodium channels open up and a lot of sodium rushing into the cell. And then our fourth change is going to be when our potassium channels open up. And this is called the falling phase of the action potential. So here we had depolarization. In here we have repolarization occurring. So repolarization means that potassium is leaving the cell through those potassium channels being open. And you can see here the sodium channels, the light gray ones, have closed their gates. So no more sodium is entering the cell. And then